Just because of time, last time, especially the first lecture, I started late because of look, somebody was late in his lecture, so I couldn't finish. So if I didn't finish for one group, I cannot finish for the other, so I had to postpone everything today. So yes, today we'll be talking about epithelium. This is the second and last lecture about epithelium. These are the learning objectives for today's lecture. I showed this slide last lecture, which is some of the features of epithelial cells. We've discussed many of those. This particular one, which is the cohesive nature of epithelium, which is maintained by certain adhesion and junction complexes. You looked in the lab this week about on some epithelial slides. This is the only request I ask. Don't talk when I'm talking, so please restrict me. Because I forget, actually, when people talk, I forget what I was going to say, because I don't prepare the lecture mentally, what I just say. So when you talk, I get distracted, and sometimes I don't remember what I was supposed to say. So you looked at the epithelial cells, the epithelial tissue in the lab with Dr. Shahid, at least some of you, not all of you. But you notice that the cells are very close to each other, and this is what we described. So it's not only because of the physical proximity, there's also something that holding these cells together. So these cells are attached together. So if you look at your position in the classroom, so you're individually sitting, but now imagine that everybody's holding the hand, to the next person. This is how the epithelial cells are. So epithelial cells adhere really strongly to the neighbors, but also to the ground, which is the base membrane. We have several structures that we'll be describing today that help with this adhesion, connection. Although we said this is a major feature of epithelial tissue, but also the junction exists in other tissues as well. And we'll talk about that this one. And we'll talk about that later when we come to other tissues. We classify the types of epithelial junctions into three main categories. The first one is called type or occluding junction. The second group, which is multiple types of junctions, is called anchoring junctions. These are cell to cell or cell to extracellular environment or base membrane. The third one, we call it, the group is called gap junctions. Although the name junction, but it's not actually function as adherent job, but rather just communication as we will see. So start with the first one called tight junctions or occluding junction. This is the most apical type of junction. So if you look at the epithelial cells, the one that's close to the apex or the top of the cell would be the tight junctions. It's not a continuous seal, but rather focal adhesion. So if you have a poster I want to seal to the wall, I can glue the whole back and then stick it. But also I can glue certain spots and then can stick it to stick well. So this is called the focal or the point of contact where the glue is there, sits on the panel. So that's why they said it is not a continuous seal, it's a continuous junction, but not continuous seal. It is there, but it's not sealed all the way. It's actually is apparent in this electron microscopic image. So the whole zone here is the tight junction, but you can see those lines. This is the one that areas actually stick to the other junction on the other cell. That's why it's not really continuous. So the whole thing is really sticking out to the other cell, but rather certain parts. The function of the tight junctions is to separate the extracellular environment from intracellular. So it is actually make sure that nothing will go in the intercellular space. Nothing is going to come, let's say, about your skin. Nothing is going to come from outside, pass through the epithelial cells and go inside to the subcutaneous or the connective tissue underneath, because have this tight band around this. So it's all sealed. So nothing will penetrate, protecting the intercellular space. The main transmembrane proteins that making these tight junctions are the occludins, and junction adhesion molecule or JAM. We'll talk about those next slide. So this is the molecular structure of the tight junction. This is the cell membrane. As you can see, the bilayer. This is what Dr. Shahid told you last week. And you can see the components of the tight junction. The occludin and occludin proteins are these two structures here. Each one has a 4 transmembrane domain. The transmembrane is the structure that crosses the membrane all the way from the outside to the outside. to the inside. It has four, each one of those included have four transmembrane domain, which is those spiral structures here. Each one of those has two extracellular loops, as you can see, this one and this one is extracellular. And the JAM, the junction adhesion molecule, which is only single transmembrane molecule. All those three, the JAM and occludin, attach to other proteins intracellularly. This is called ZO1, ZO2, and ZO3. The ZO1, ZO2, or the ZO1 abbreviation stands for zone occludens. This was the old name they gave it to the tight junction, so they still use this for the protein name. So ZO1, which is the big blue one here, and then you have ZO2, the gray, and ZO3. They interact with each other. ZO1 mainly, after that, bind to the actin filament. So this junction not only attached to each other, but also they have the intracellular domain attached, which is ZO1 and ZO2, attached to the actin filaments. So they're anchoring inside. When you want to have something really, hold really well, you actually have something to anchor it outside. The wedges that you pop at the top, they put it in the wall, you actually can have it anchored inside. So when you try to pull it, it doesn't break it because the actin filament is attached to it, so you can't get it. So just a clinical note here. The H. pylori is a bacteria affect the GI system, causing some diseases. These bacteria secrete toxins and go inside the body, secrete toxins that breaks those tight junctions. So what happened? Now we have the area between the cells open. Things can go from outside to inside. That's why those bacteria, once they break the tight junction, people get ulcer because the acidic environment in the stomach is supposed to be sealed, that doesn't go inside. Now it can seep in. So to destroy the tissue underneath and causing the ulcer. So just a hint of why we study something like tight junction. Okay, it's not important. Other types of junctions called adherent junctions. Yes.
My question was, what's the difference between closing and unclosing?
What's the difference? Just two different structures of the protein. I mean, the different molecules that making the.
Do they have a different function or same?
Well, it's not known. It's part of the whole. Yeah. At least I don't know. It might be some research on this, but I don't know. So the other type is called adherent junction. This is the junction that's underneath the tight junction. So the first one that's close to the lipids. Yeah.
Can you please go back one slide? It's in the jewel itself is the membrane.
The cell membrane? Here's the plasma membrane. With one cell, this intracellular space. This is transmembrane. So this extracellular and this intracellular. The adherent junction, which is another type of junction below the tight junction. The first one, tight junction, followed by the adherent junction. They anchor the cells to the neighbor. This one hold really tight the cells together. The other one was really sealing the area, but this one grabbing the other cell really tight. Mediated by what we call cadherin, a transmembrane glycoprotein that we describe next slide. And here it is. Again, this is cell membrane here, cell membrane. This one cell, part of one cell, this one part of one cell. Now they have to attach together. There is an expression of this E-cadherin, a transmembrane protein, the yellow one here, attached intracellularly to the catenin, which is the disc, and the catenin attached to the actin filament, again anchored inside. So I have the domain, the transmembrane domain, attached to the intracellular part, which through the catenin and the actin filament. Something important about this area where actually have the actin filament close to the membrane, they call it the terminal web. Dr. Shahid talked to you about the cytoskeleton. There's special arrangement called to certain areas where actually those filaments getting really dense. They call it the terminal web because the structure of the web. They are very, very attached to each other and giving where these adhesion molecules attach. The calcium ions are important to make this connection between the two different adherent molecules to form the junction. The second type is called desmosomes. This and another one also the junction. Let's hold the cells together through what we call the plaque, desmosomal plaque, which is this disc structure. Again, this is the cell membrane, this cell membrane, this intercellular. You have a disc structure called desmosomal attachment, and attached to the intermediate filament inside the cell. Outside, we have those two molecules, the desmoglin and desmocolins, which are these two bars here. attached to each other from the other side through calcium-dependent adhesion molecules. They need calcium also for this adhesion. Interestingly, we don't usually see the tight junction in the HND preparation, but the desmosomes, sometimes you can see them like this one in the skin. Sometimes it's some preparation, you can see those desmosomes and this thin line. Focal adhesions. Now we talked about three types of adhesion that attach the cells together: the tight junction, adhering junction, and the desmosome holding the cells. Now we said also the cells, the epithelial cells attached to the underneath, which is the base membrane. This one of them, the focal adhesion. So this structure link the cytoskeleton. So three components: intracellular, transmembrane, and extracellular. Here, this is the cell membrane here now this time. So the cell is up here. So this part is inside the cell, this intracellular compartment. This is the base membrane, correct? So they're anchored inside the base membrane. So three components: one inside the cell, which is made of this cytoplasmic component, have this paxillin, talin, vinculin, all these molecules attached to the alpha actinin, the part of the cytoskeleton. Then a transmembrane integrin. I mentioned that yesterday or last lecture. I said the integrin, anchor those the basal part of the epithelial cells to the base membrane. And then when you have this focal adhesion, those integrin attached to fibronectin, which attach it to the rest of the basal lamina here. There's another junction that holds the cell to the base membrane. It's called hemidesmosome. So focal adhesion and hemidesmosome attach the cell to the extracellular matrix. Again, provide more adhesion to the basal lamina. This is particularly important and present in an area where we have friction. Your hand, for example, you will find the skin here have more of those desmosomes, hemidesmosomes. Why? Because you want to hold the skin tight to the underneath. Otherwise, you can have blisters. Right? And some people have disease that have genetic defect, and those junctions are not really strong. So every time they work with something, they get blisters all over their hands. Normally, you shouldn't, unless you work like when an axe for a couple of hours, and you never do, then you get blisters. But some people with this disease, they can blister easily because these junctions are not there. So you can always break. Anytime you touch something, you break the epithelium from the underneath. And that's why you get the blisters, actually, because the feel good in. Again, it's very similar to focal adhesion, just the proteins are different. Plectin, the PP230, and erbin making the intracellular compartment. And then you have also the integrin here, the same, and then attached to the laminin type 5 in the basement membrane. Gap junction is the last one. Again, although you call it junction, but it's not actually a junction. It's a communication. It's a channel of communication between cells, as we'll see. It's made by this unit called connexin, and multiple of those connexin make a shape called connexon. So imagine the connexin is your finger. So I have 5 fingers. This will be connexon. This is the whole unit. So you have a pen or pencil and put 6 pencils actually together, make this unit will be connexon. So the unit will be connexin. Connexon is the bulk. And they span the cell membrane. Again, they are transmembrane. They cross the whole membrane. Each one of those connexin made of 6 connexin with a central canal open inside them. As you see here. So this one layer, cell membrane. This is a connexin. So this is a gap junction and this is a gap junction, and they connect. And here it is in this illustration again. You can see there's one and there's one, and they align. This is the main function. You have one tube and then one tube. So it's almost like, as I said in this example, if you want to go, if you have, let's say there's a next room and there's another room next to us. So when you try to leave from this room, you go through outside the main door, you go to the other room, go to the main door. But if we break through the wall and make another wall, this will be much faster for us, correct? Because we can just channel. It's exactly the same function of the gap junction. Cells in certain situation, they need the communication to be quick. So we don't have to release a signal outside and then the signal go to the next cell and the signal respond. No, just shoot this through this tube, which is the gap junction. And you can see in this arrow how this is actually communicating. So once they align the connexin, which is this group of connexin, opposing the plasma membrane, they form this complete intercellular channel. Did you see them how they align the pipes? If you ever looked at this, they bring the pipes and try to align them until they fit, and then they tie them together. This is exactly how they connect them. One pipe, another one, and you align and they connect, and then the channel the canal or the pipes are connected. And you can see again this image, one here, gap junction aligned with the other one. Again, we usually see those in clusters. You call it the gap junction plates. It's not individual ones scattered, but rather have 50, 100 of them clustered together. And they can be visualized with electron microscope. You can see this area, all these little dots are the gap junctions coming together. This is the only way we know so far that actually permit direct passage of signaling molecules from one cell to the other. Otherwise, that's a literally stay outside. It's the only way that you can communicate. And again, although we said it's actually described epithelium, it's present in other tissues. One of those is actually the cardiac muscle, for example, when Dr. Shahid will talk to you about this. The gap junctions are present there because, again, we need to signal propagation, the electric signal to go between all the muscles to contract at the same time. So the fastest way to send this is through the gap junction. So we talked about the polarization of the surface of the epithelial cells, and we said there's apical surface always. Some of those have special features that increase the surface or do different functions. These are structures called microvilli, cilia, and stereocilia. We start what we call the microvilli. These are finger-like cytoplasmic projection. So instead of the cytoplasm or the plasma membrane is straight line, it actually make projection. It goes up and down, up and down. So it increases surface big time. Here it's a histological image showing those microvilli on top of this cells. What kind of cell? What type of epithelium is it? Simple. Yeah, and these are the microvilli. Okay, Dr. Shahid will show you some slides, but will never be as good as this one. No, just because the type of microvilli, yeah, we didn't pay enough money to. Yeah. So this oil immersion lens. So this high magnification and good quality preparation. Most of the time, it's going to be a fuzzy border on top of the cell. It's not going to be really clear as this one. When we look with the electron microscopy, you can see this cytoplasmic projection. These are the microvilli. This is a nice picture where you can see them all similar. They're about 1 micron in length and about 0.1 micron in width. So they're really tiny structures. And you can see here in this electron microscopy how they look like, finger-like projections. But there is a variation in their shape. Sometimes, stop talking. The one on my left, I see you, and I know you also. So please don't talk. So these are the shapes of the cell can vary. You see, I forget when I start talking like this. Yeah, the shapes can vary. Sometimes they look like ball-like structure or blade-like instead of really pointing finger. I'd rather have you leave if you're going to talk. And the function of this microvilli usually cover the absorptive areas, like in the GI. So you always know from physics that the straight line is the shortest distance between input points. The fastest you want to go, you put a straight line. But if you want to delay things, you make it wavy. So this is exactly what happened with the microvilli. Instead of the straight, you finish it. Then the membrane goes like this, like that. And we need that in the absorption because we need to slow down the process. Otherwise, the food will go quickly. Yes.
Is there a difference between microvilli and villi?
Yes. That's a big difference. But it's not the time. So, like I mentioned in the beginning, the last issue you mentioned, it was related to the GI. Yes. But not this different. The microvilli are structured on the cell. The villi actually a group of cells making different much bigger. Okay. So in the GI, we need to keep the food long time. So we delay it by making this road. So if want you to take a long time, I'll take this route. But if you take it instead of the highway, you're going to be going inside all this side streets will take you forever. So in the GI, we need that because we need food to stay for a longer time. And actually, there's certain diseases where the people lose the microvilli and the villi, and then they develop certain diseases. So we look at the outer structure of the microvilli. So each microvillus have this core of actin filament capped with certain protein called actin capping, which is those light green ones here, and they attach it again to the terminal web in the part of the cytoskeleton. The second structure called stereocilia that's also present on the apical part. It's much less common than microvilli or cilia. They can be found on some absorptive epithelial cells and also in the male reproductive system. They increase the surface area and also facilitate absorption to some areas, resemble microvilli containing also actin and the same ultrastructure binding protein, and they attach to the web terminal. So here how they look like. They're bigger than the microvilli. They're not as dense. Yeah, the microvilli, we talk about that. The stereocilia are typically much longer and less motile than the microvilli. The last structure is the cilia. This you will see well in the with Dr. Shen. Highly motile apical structure also larger and longer than the microvilli. So the microvilli is about 1 micron tall. This is 10 microns, so it's much bigger. So it's almost as tall as a cell or half. So these are hair-like structures. They wave and they move. That's why they call it the hair-like structures. The microvilli, sometimes they call it brush border. In the old days, when I used to have hair, time ago, we used to have those brushes that have short but very dense hair. I don't know if they still exist or not. I don't use them anymore. But the hair because they're very tight, they're short, and they're next to each other. So they carry the brush border. This is the microvilli. But the cilia, they call it the hair-like structure because like girls' hair waves, it's long. But that's why they give the different name. Yeah, I always enjoy mentioning that I used to have hair. They look different, by the way. Yes. What? The cilia exist in certain part of the body and the stereocilia exist in certain. They have similar function to some extent. But because of the density of the cilia, usually the motility is very important for this more than anything like absorption. The stereocilia mostly for absorption and sometimes for motility of the body. So the cilia are present mostly in things like columnar epithelium, sometimes cuboidal epithelium. You can see a very nice illustration here. Again, you'll not see anything like that in the lab, but similar, close maybe. And this scanning electron microscopy, we're looking at the surface now, so you can see how wavy those cilia are. They divide them into different types. Motile cilia, they call it, which is the common. Most of the cilia are called motile cilia. There's something present in the embryonic cells called nodal cilia, and then primary cilia, which is usually single cilia, present in most of the cells. And sometimes people confuse the primary cilia with the stereocilia. It's because also sometimes the stereocilia is present as individual, one or two only. When we look at some of the preparation, the surgical preparation, we look at the cilia, which is the line here. You can see another line, which is the top of the cells, but it's very dense. They call it the basal body, and we'll describe that. But you see this staining here, they call it basal body, which is where the cilia anchored in the cytoplasm. We mentioned that in details later. Ultrastructure of the cilia. So if we have a section of the cilia, cross section, and we look inside, what are the molecules and ultrastructure in there? It's made of core, which is 9 peripheral microtubule doublets. So this Dr. Shahid covered microtubule. I know he did. So we have 9 doublets, 9 groups. Each group is 2 groups. Each one is a microtubule, 1 group. So 9 by 2 is 18. And then we have 2 central ones. They call it the 9-2 assembly of microtubules. The whole structure is called the axoneme. So we have microtubules, 2 of those come together, and then we have 9 at the periphery and 2 at the center. The axoneme, which is a structure, those beams that making the cilia, attached to another structure, which is the basal body that we mentioned, made also by microtubule, but these are in triplet. So you're almost anchoring a pipe or a tube into another tube. So these are the microtubules from the cilia attached to a microtubule structure inside the cytoplasm. So in this illustration, showing you a little bit of details. So these are the doublet. So each one of those is made of microtubule. Each one of those microtubule is made of those little structure that Dr. Shahid told you. Anybody remember what's the name of this? Which one?
Microtubule.
See, this is microtubule. Yes. There's a little thing here. They're making the microtubule.
Microfilaments.
Protocol. Protofilaments. The protofilaments is made of what?
Alpha exactly.
It's a better use. Yeah. Oh, you remember. That's good. Yeah, it's some good. Yeah. So this is making the microtubule, and then the microtubule will come. When you look at the structure of those doublet, we said the microtubule is made of those 13 protofilaments. Right? But they actually, because they're joined, they share 4 in the middle. So this actually, if you count 13, and then total will be 9, not 26, because you share. Like one was split circle. If you look at this picture, they're sharing. And the other structure you need to know, which is the nexin, which is the linking protein, because those microtubules connect together. And also, there's other things about the dynein arm. It's helpful to contraction. You don't need to about the mechanism. But notice that the cilia is motile. So this thing actually with the dynein arms, they make a click move. So they wave because they said the beating move of the cilia. They wave in one direction and then beat, and then they move and they beat.
So microtubule A and B both share the 13 protofilaments. Is that what you mean?
No. They share this.
The one in the middle.
Yeah, this one is complete, 15, but this one is 9 because the borrow this. So if you count, it's not going to be 13 and 15. But it's individually, if you look at this would be 13. This would be 15. They share some of them. Now, glandular epithelium. So one of the things that epithelium does, it actually has what we call secretion. Secretion means that you release some contents from inside the cell outside, but you secrete something. The secretory cells inside epithelial cells can be actually individual cells scattered between other cells that doesn't secrete. Let's say you have 100 students here. Their function probably is to major in medicine. But maybe 10 of you scattered among you, you also do business degree as well. So they have a different function compared to the other one. You don't study only medicine, but something else. So some of these are scattered. These are individual secretory cells. But also they can come and compromise organ. So some of those, cells, they double major medicine and business. They come together and make another group in a different class, but they're all the same double major. So this is exactly how the secretory cells are. They can be scattered among other cells that has nothing to do with the function. But in the meantime, also they can group together and have their own function. In this case, they call it gland. They have a different name. Now we call them the double major medical business students. So it's just a histology of some of this. Dr. Shahid will show you and explain that in detail later, but these are glandular structures with the duct, as we'll see. How the gland development, during development, epithelial cells will grow inside, making this structure, like here, we multiply, and then either we have a continuation with the outside through a duct, or they'll be totally separate from the original epithelium. When they have a duct connected to the outside, we call it exocrine gland. This is the type of the gland. If it's totally separated, we call it endocrine gland. The difference between the exocrine and the endocrine gland, that exocrine gland secrete the products through a duct. So the gland will make a secretion, the secretion will be selected through a duct, and the duct will release the contents to the outside. Endocrine gland, there is no duct because they're disconnected. They're just a bunch of cells coming together, but they secrete something. Where this secretion will go? It will go inside the connective tissue and then will be absorbed and goes inside the blood or the lymphatic tissue, mostly the blood. So it goes in a product that we call hormones. So your sweat gland is an exocrine gland because the sweat come out on your skin. You feel that. But do you know about what happened? Your thyroid is a gland, your pancreas is a gland. Do you see anything coming from your neck? It's also superficial. I mean, it's right underneath the skin. But you never anything because it's an endocrine gland. It releases the contents that goes inside the blood. It has an important function through the blood. So this is an endocrine gland versus exocrine. Exocrine gland is what we focus about here. The endocrine will be covered mostly whatever endocrine block. Endoreproductive. Yeah, and sometimes with the epithelial cells, they secrete the substance that doesn't go inside the blood with the endocrine gland, but rather affect the other cells neighboring. We call it paracrine effect. So the component of the exocrine gland, they have two components, the part that secrete and the part that carry, the secretory portion and the excretory duct. So if you look at this illustration, this yellow is the part that actually functionally secreting something, like the contents, and this carried by this duct to the outside. So this is the duct and this is the secretory portion. Again, this is an example of the cells that I said these are the double major. They do different things. They're epithelial cells, but also they secrete. So you can see scattered among other cells. The one that's in purple, they don't do the same. These are only the one that make the secretion. So when you have individual cells among other cells, we call it unicellular gland or goblet cells. When you have those glandular structure, we call it the multicellular gland. So another histological image, you can see these goblet cells, and you can see in this nice one, the secretion goes directly to the surface. No duct, nothing. One cell just excrete things. And here you can see this multicellular, all the cells are glands, go inside the duct, and then secrete the content. So if you look at the whole gland, it has a real function. So these are the individual units, and each one has a duct. And then you can see all these little things are individual units, and then they have one duct, and then they join a bigger duct and a bigger duct. This is similar to the grapes, the inab. When you look at the grapes, each one of those has a small stem. And the stems are actually connected to the bigger stem, and then the bigger stem attached to the one. And so it's exactly how the gland. These are the gland, the secretory units, secrete inside small duct. And then multiple ducts will come and secrete in a bigger one, and then a bigger one will join a bigger one until you got the main excretory duct that takes things outside. Usually it's single. So it goes, and then have connective tissue in between them, plus dividing them. Classification of the multicellular, they have different shapes. The ones either long, we will call it tubular, or rounded, which is called acinar. And it depends on the number of branches. So if it is simple tubular, it has only one unit. If it's more than one, it's all tubular, but they all open in one duct. It's called be simple, a simple branch, or it can be coiled. Can be also acinar. It looks funny here. It doesn't look rounded. That's supposed to be rounded. But this is simple acinar, and then simple branched acinar, which is branched but acinar. And then you have a compound, which is actually going to have multiple ducts to open to the main one. So have 1, 2, 3, and open to the main one. Here, 1, 2, 3, and each one can be tubular, the unit, or can be acinar, rounded, or can be mixed. And a lot of those cells will be mixed. Notice that when Dr. Shahid will show you some of the slides, and we said, okay, there's a tubular gland here. You have to cut in luminous section to see the tubular, because if you cut in cross section, it will look like the acinar. So say, oh, I don't see how you said there's a tubular gland here, but no, you have to look for the one that actually will cut in the luminous section. The acinar doesn't matter, because if you cut the ball in any dimension, it's always rounded, but the tube is not. If I cut it like that, it's going to be longitudinal. If I cut like this, it's going to be cross.
Say people, like when I look under a microscope, how would I differentiate between branched tubular and branched acinar? What's the same?
No, I'm not going to require you. Just tell you how you can exit like this. It's not always easy to differentiate. Now, how the excretory gland secrete? There are different mechanisms of secretion. The first one is called merocrine secretion. The secretory delivered in a membrane-bound vesicle to the apical surface. So the contents are packaged in a vesicle with a membrane and shipped through certain mechanism to the apical part. And then it joins the cell membrane and then releases the contents outside. This is called merocrine secretion. And this is the vesicle actually fused with the membrane, and the process called exocytosis. You come to the membrane, fuse with the membrane, and release the contents, called exocytosis. And this is the most common way of releasing mechanism for the excretory gland. The second one is called apocrine secretion. It's very similar. Also the package, but usually the package is big this time. That's why you find this in things like the lactating mammary gland, big lipid droplet. So it's also packaged. But when it comes, it doesn't fuse with the membrane open, but rather take part of the membrane and release. And one of the students said, okay, but if all this vesicle come and take part of the membrane disappear, yes, but there is a replacement mechanism. It's like you're almost cutting. It's like the dough. You remember when somebody making the bread or طعمية, and the packer one must as a man can you hold the طعمية and then you keep cutting until it disappears. But yeah, the same thing. But then he replaces it with something. Then yeah, طعمية like طعمية. The holocrine secretion. This is the third part. The secretory products accumulate within the cell, and then the cell basically bursts. It just commits suicide. Here it is. You release everything, and the cell dies, and there's a debris. Here it is. So When the time comes, the cells are filled with the secretion, then the cells burst, the membrane breaks, contains rays, and the cell basically dies.
Is it related to apoptosis?
It's programmed cell death. It's not because it's different. There's it's certain things, but it is part of the programmed cell death. There is a fine difference between the two. So this happened in the sebaceous gland. You remember, especially our faces, inside all the sebum, the white secretion, and you press on it. This usually it's a holocrene. You break this thing. When it comes to contents, you will find the contents either what we call serous, which is basically synthesized protein inside, mostly not glycosylated, they're just enzymes, or mucus. Goblet cells, for example, use filled with mucus, and you can see this in the histological specimen. Serous secretions in this gland, you can see it's usually going to be dark and purplish color. The mucus is clear because of the perforation and the fact that it's actually clear solution. So Dr. Shad will show you this. There's some difference between them. Renewal of epithelial cells. Somebody asked about stem cells before. I think it was a girl. But stem cells is the cell that can divide to another cell, so they give birth to another cell. So during embryonic life, early in development, the cells they call it non-committed. The cells doesn't have any particular function yet. So it can be brain, can be heart, can be skin. Right? This is very, very early development. But with specialization now, they become tissue. Now the stem cell inside each tissue have a function to divide to the same type of tissue. So if you have stem cell in the epithelium, it's not going to make a brain cell. It's going to be only because they're already committed. They went through several stages. It's like you're already in medical school. You're not going to take engineering classes. You're already committed to this. So what you take is engineering class. Yeah, you can teach to other medical students, you can teach to other doctors or so on. But now I can't talk about engineering. People have done all my life, something different. So the same thing with this. These are committed to certain career, but the earlier development is different. Epithelial cells have the ability to regenerate even compared to other cells much better than connective tissue or muscle or brain, even if they have any. So that's why you remember if you get a skin wound, you heal well. Same epithelium, see, not only other tissues. When people get heart attack or cerebral ischemia and the brain tissue dies, you don't replace it. That's it. It's done deal. You can't replace it because you don't have stimulus. The muscle doesn't have ability to regenerate at this particular tissue or this particular organ, so you end up with this. So all the research now is trying to find an early cell that you can actually grow in the lab that can commit to other things. So I can replace the heart muscle that was died because of the heart attack with a new muscle in the brain and the cartilage and so on. Skin, nobody cares, because it regenerates most of the time, that's easy. Again, if you look at your GI, we replace our epithelium all the time. We slap epithelium all the time. Just to regenerate. You don't feel even anything happen. But with the tissue there. And I mentioned that because there is a guy called Hal Broxmeyer, who's a professor of School of Medicine, IU, he was from New York. He discovered actually that you can separate the stem cell from umbilical cord. So since at that time they collect all the placentas and umbilical cord from any woman that come, they don't know what to do with this. Yes, just researching, but they think there's stem cells that can differentiate. That they keep banking those cells. But eventually, somebody can find a way for those to be. So diseases. So again, for the 150 time, I think I say that. I'm not going to ask you about disease. You're not supposed to know disease now. I just give you the example to just link it to the basic science. So because I've been working on cancer biology for all my life, basically. So I usually like to give an example of cancer. So cancer is the cells divide uncontrollably. You just keep dividing rapidly. There's no control. They don't know when to stop. So if you look at this, picture cancer cells, which is supposed to be normal here, they start dividing when they have different genetic mutation, of course. And the thing is with the cancer can happen at any kind of tissue. Cancer can arise from epithelium, from connective tissue, from muscle tissue, and also from the nervous tissue. But almost 90% of all cancer coming from epithelium, we call it carcinoma. That's why it's important to know this. Again, for several reasons. One of them is exposure. Just everything. I mean, your skin is exposed to all kind of junk. Your GI is also exposed. So the chance, because also the ability of the cell to renew, that because we are dividing, the chance of actually mutation errors is higher, so people can develop. Yes.
It's like that malignant tumors can arise from most types of epithelial cells. Can benign tumors also arise from epithelial cells?
Yeah. Any place can have a benign or tumor. So again, because of most of the time when you see somebody with a cancer, it's going to be carcinoma. It's going to be coming from the breast cancer, prostate cancer, all these are coming, urinary tract cancer, all this coming from epithelial cells. And sarcomas actually is what we call connective tissue and so on. So the other thing also you have to notice about this is we talked about the basement membrane, correct? And we said that epithelial cells shouldn't pass through the membrane, but it will in the case of that. You break through it. And pathologically, when you give the sample to a pathologist to look at and you see all the cancer cells there, but they already invaded the basement membrane, it's a bad news for the patient. Because remember we said the epithelium doesn't have blood supply, doesn't have lymphatic. But in the tissue underneath, there is. So once it break in, it have access to the circulation. And now they metastasize. Metastasis means spread. Now the cancer that was supposed to be localized in the breast or the prostate, it breaks through, it went through circulation, and goes anywhere. That's why a problem with the cancer is not the tumor itself, it is the metastasis. Most of the patients die because of the spread. They remove the cancer from the prostate, from the breast, from the lung. 5 years later, you find the cancer in the brain and in the bone. And this is the problem with the cancer. Because few cells just escape this membrane and are able to penetrate through the blood. So that's why important. This membrane is one of the things that clinically really important, probably the most important thing in the whole histology class. But anyway, so other disease also cilia. We said the cilia is motile, keep moving things, because moving mucus, for example, from the respiratory system. If it's not moving, there's a big problem for a patient. Some people have genetic mutation, we call it Carter-Jenner syndrome, immotile cilia syndrome. The cilia doesn't beat, it doesn't pick up. So all the secretions coming in your lungs, nothing can move it up, it sticks there. So they get infection and trouble. These kids get really sick because of it starting really early in life. Again, some of those people if they live long enough, they're infertile because all this motility is not there to move their sperms and so on. Well, this three lectures back to back. I'm like dying. Yeah. Anyway, so this is the way you should study. Put things next to each other. Because otherwise, next week, when we talk about histology of the connective tissue, Dr. Shah will talk about the muscle and so on, everything will look the same. Everything is like blue and red. So try to put certain things together and put your own, key. Because especially with the OSPE exam, and OSPE exam actually have a quite relative good percentage on your grade, and it's easy. So make sure that you have your own clue. I know this because of this. I know I can differentiate. If you sit and think on the image or the slide that Dr. Shahid will put, not me, or him, and you start thinking, you're probably not going to get it. If you don't go exactly what I'm looking at, if you don't look at the epithelium and say, why you know this epithelium? Well, because the cells are crowded. It's not connective tissue. Cells are next to each other. If you're still thinking about what is this? No, there's no time. You have to know. That's why you have to practice, put things together. What's the difference between this? What's the difference between the epithelium and the connective tissue? What's the difference between the connective tissue and the muscle? How do I know this? This is what gets you in the exam. See, because you don't have a key, you don't have something in mind that I use this criteria to differentiate between this and this. You just randomly try to find it. No, there's no time. You're not going to find it. I give you another half an hour, you're not going to find it. It's still going to miss it because you didn't know what it is. You have to be prepared for this. The same also with the exam, with the written exam, which I put mostly, or Dr. Jared also put some. But you have to have a key. I mean, you're looking at the question, it's simple. But there is a key there that if you don't know what you're looking at, if you don't know what the classification of epithelium, if you don't know what the name, then you're in trouble. So try to do that early, and you don't have much. I mean, probably by next week we'll be putting questions, for the final exam. Thank you. Take your attendance. And thank you for being quiet at the end. So that's